So we were discussing about DNA sequencing. So we know that there is an enzymatic procedure for DNA sequencing that is called a Sanger method or dideoxy sequencing method. The principle behind this is that the DDNTP will stop or terminate the sequencing. So the termination of DNA synthesis is the most vital feature of dideoxynucleotide sequencing method. We have discussed the principle earlier. So the DNA, we know that DNA is having a double helical structure. So it is having a double helical structure. Will be having two polynucleotide chains. So we need to separate the strands. We need to separate the strands before sequencing. So we use what will we use? We use temperature for denaturation. We already know that we have done in PCR. We have done in PCR to separate the strands. We used 95 Celsius there. So the same technique we are using here also. We enhance the temperature, then the double strand will separate and we get single stranded DNA. So for and another thing we need is the for polymerization we need an enzyme. We have already studied about Taq polymerase that can withstand the temperatures, changing temperatures, that is called a Taq polymerase. Another type of enzyme we use for sequencing is sequenase. It is sequenase obtained from phage T7. Phages, you know that bacteriophages are viruses. So the type of phage that is producing sequenase is phage T7. So we need what? A polymerase. Then we need a primer. We know that to start a polymerization we need primers. What are primers? Primers are oligonucleotide chains. Oligonucleotides means a few nucleotide length sequences. The primers will be having about uh, 10 or 25 nucleotide length. Here complementary to the template strand. So here will be C or G or T, A, like that. Here you can add. These are synthesized, chemically synthesized. Primers are actually chemically synthesized that are complementary to the template strand. So we require a primer to initiate the DNA synthesis on a single stranded template. So these are chemically synthesized sequences, about 17 to 24 nucleotides that are complementary to the template strand. So it binds to, primers will bind to the template strand, and that will provide a 3' end, that will provide a 3' OH group, so that polymerase can add new nucleotides here. So the function of primer is they will give a OH group, 3' OH group, so the polymerization can be started. The primer provide a 3' hydroxyl group for the initiation of DNA synthesis. And so we need in DNA sequencing, in conventional type of DNA sequencing, we need, that is Sanger method, we need what? 4 separate reaction tube. Reaction tube will be like this. We need 4 different reaction tube. These are reaction tubes. So in the reaction tubes, what we have to add? We have to add template DNA. We have to add template DNA in single stranded form. We have to add in 4 reaction tubes. We have to add template DNA. So we need to add template DNA here, here and here. So next we have to add DNA polymerase. Polymerase we need to add. We need to add DNA polymerase here also. Then we have to add primer. Primer is also important. Plus primer, plus primer, plus primer. Next we have to add 4-deoxynucleotide triphosphate DNTPs. What are the DNTPs? DATP, DGTP, DCTP and DTTP. DTTP. So we need to add these 4 numbers here, here, here and here. So in this reaction too, you need to add template DNA, primer, then polymerase enzyme and these DNTPs. And here only one difference is that you need to add one of the 4 modified nucleotide. What is the modified nucleotide? Here is the modified nucleotide. So we need to add only one of the 4. We have 4 different types of DDNTPs. What are the DDNTPs? DDATP,